Everyone, this is Soumit. I'm pursuing my master's in RVSKVV University from India. Now I'm going to present about my investigation, that is study of dust characterization and diversity analysis in advanced breeding lines in chickpea crop. Generally, the main aim of plant breeding is to develop improved varieties of crop plants. In order to release a variety, some characteristics are more important, such as distinctiveness, uniformity, and stability. This is called as dust. And dust test, at least it should be done for once for the varietal release. Now the dust was changed to N dust. N represents novelty. And genetic diversity is nothing but it is a variability present within the species among the traits present in the species. And this genetic diversity, it is very much helpful for the genetic improvement of the variety. And as you know that India is the second most populous country, due to increase in the population, we should produce more and more amount of food production, that means crops, to sustain ourselves, because here the total export and import share of chickpea in India was 148 and 152. The import share was more than export, so we should improve our production in crops. The objectives of my investigation was to assess genetic variability of genotypes through dust descriptors, which was given by UPOV, and it helps us to produce genotypes which enhance its crop yield and quality. And second objective was to study the genetic diversity among the chickpea genotypes. High diversity will reduce the losses in crop production, thereby it will improve the quality and production of the crop. Methods. The present investigation was conducted at research area of All India Coordinated Research Project of at RK College of Agriculture, Sihor, Madhya Pradesh, in the season of Rabi 2021 and 22. In this, what I done was categorization of genotypes according to the dust descriptor, which was given by UPOV under the guidelines of Plant Protection Variety Farmers' Right Act given by Government of India in 2001. Number of genotypes which I took for investigation was 120, and which was laid out in a design augmented with a row length of 11 meters and row to row distance 30 centimeters and plant to plant 10 centimeter. Date of sowing of my crop was November 20, 2021. Statistical analysis done in this research was genetic diversity analysis. Total of 20 characters were studied for dust characterization, and the 120 genotypes were allotted according to the observed characters. The characters observed were anthocyanin pigmentation, heighted initiation of first flower, time of flowering, plant growth habit, color of foliage, leaflet size, leaf pattern, flower number per peduncle, flower color, stripes on the standard, peduncle length, plant height, pod size, number of seeds per pod, color of seed, seed size, seed shape, seed texture, seed ribbing, and seed type. Among the 120 genotypes, 113 genotypes are of Desi and 7 genotypes are of Kabuli. Highly significant variability was observed among 120 genotypes and grouped into these categories. Among these 120 genotypes, Dindori was one genotype which is of Desi type but shows Kabuli characters, such as Kabuli type in which the flower color will be white. It is characteristic of white flower and creamy beige seed coat color, and without anthocyanin pigmentation. In Dindori, it is of white flower instead of pink and without anthocyanin pigmentation. RVKG 2K21 is the another genotype which is Kabuli, but it shows desi characters such as pink color flower and with anthocyanin pigmentation. Genetic diversity analysis among all 120 genotypes, these are categorized into 11 clusters. Among these 11 clusters, cluster 2 is with maximum number of genotypes, 30, and cluster 11 was with 2 genotypes. Among these 120 genotypes, ICC 10401, ICC 14205, RVSSG 116, RVSSG 122, RVSSG 125, and RVG 202 record high cluster mix, in which that means high amount of variability was present in these genotypes, and they can be further used. Recombination breeding in between these genotypes from all these clusters, it will be very useful to increase the recombinant lines. Thereby, it can increase seed yield and yield contributing characters. That means in quality. Suggestions for further work. Based on my research, I can say that it is of 6 months of research, but these morphological dust traits can act as a physiological phenotypic markers, which can be very much useful in varietal identification, and this can be further tested in different environments too. Genetic variability and characterization can be used in breeding programs in stress-prone areas also. Thereby, by this multi-locational testing, we can evolve stable. We can emerge stable varieties, and recombination breeding of these genotypes in further breeding programs will be helpful for development of new plant varieties with highly significant characters. By this, I will end my presentation. Thank you.